Hi, today we'll be talking about glutamine, glutamate, and GABA, and how these are all related. First off, glutamine has a structure like this. Now, although we won't actually analyze structures too much on this channel, it is important to know how glutamine will look in comparison to glutamate and GABA. A healthy adult human contains over 80 grams of free glutamine, with greater than 98% of that glutamine inside skeletal muscle cells at concentrations of around 20 millimolar and above. Glutamine itself is synthesized by the enzyme glutamine synthetase using glutamine and ammonia found in the cytosol, and this usually occurs when glutamine is released by glial cells and is metabolized to glutamate by glutaminase, which is only found in mitochondria. Glutamine itself has many functions, being a substrate for protein synthesis, a substrate for urogenesis, a substrate for hepatic gluconeogenesis, an anabolic precursor for muscle growth, an oxidative fuel for intestine cells of the immune system, interorgan nitrogen transport, acid-base balance in the kidney, precursor for nucleotide and nucleic acid synthesis, and it's a precursor for glutathione production. But what we're mainly going to focus on today is how glutamine is used for neurotransmitter synthesis. This is glutamate. In the 1930s, high concentrations of it were discovered in the brain, leading to a bunch of trials in the 1940s around dietary glutamate and glutamine to treat learning disorders and epilepsy. People originally thought that it was going to be used in energy metabolism, given the close association of the amino acid with the Krebs cycle. This is because glutamate can be synthesized by transamination of 2-oxyglutarate using glutamate dehydrogenase, an intermediate and important step of the Krebs cycle. But this all changed in 1954 when Takashi Hayashi published in the Keio Journal of Medicine the possibility of glutamate's role in the brain, after he injected people with glutamate into their brain and carotid arteries, producing actual goddamn convulsions. Glutamate itself is synthesized via the mitochondrial enzyme glutaminase, which converts glutamine into glutamate and ammonia. Nowadays, however, we know that glutamate works on a cellular level, usually by the process of glutamatergic neurotransmission. First discovered in the brain in 1950 by Eugene Roberts and Sam Frankel, nobody really cared when GABA was discovered. Its real effects were discovered in 1957 by Canadian scientists A.W. Basemore, K.A. Elliott, and E. Forey when they claimed that GABA was causing inhibiting activity in crayfish. It was noted that glycine was also important in inhibiting brain neurotransmission, but GABA is clearly the most abundant one. Researchers went to work in using it to manage epilepsy, the disorder that is obviously caused by overexcitation of brain neurons. Although it had showed promise and had worked in terms of fixing disorders caused by overexcitation, it wasn't used beyond clinical trials because, as you can imagine, tampering with the presence of the major inhibitory neurotransmitter in the synaptic cleft can have severe side effects, including but not limited to numbness, anxiety, drowsiness, etc. GABA has, however, spurred a lot of research around it and is most often recognized in the process of GABAergic neurotransmission. So overall, the glutamate-GABA cycle is the transfer of glutamine from astrocytes to neurons and neurotransmitter glutamate or GABA. After discoveries of intercellular compartmentation of glutamine and glutamate pools were made related to astrocytes and neurons respectively, it had led to the suggestion of glutamine and glutamate having some kind of cycle working between glutamatergic neurons and surrounding astrocytes. Before getting into the cycle itself, however, it's important to know why this cycle exists in the first place. Neurons themselves are incapable of synthesizing glutamate and GABA from glucose because they lack the enzyme pyruvate carboxylase. Two variations of a glutamate-GABA cycle. Of course, all three of these neurotransmitters can be linked together, but glutamate and glutamine have a different relation compared to the relation that GABA and glutamine have. In the case of glutamate, when glutamate is released into the synaptic clefts from the presynaptic terminals, one of two things can happen. It can either be reuptaken from the synaptic cleft and put back into the presynaptic neuron, or glutamate can be taken up by an astroglial cell. When in the astroglial cell, glutamate can be recycled via the Krebs cycle, or it can interact with glutamine synthetase. Using glutamate and ammonia, which usually comes from the presynaptic neuron, glutamine synthetase synthesizes glutamine. this glutamine is transported back to the presynaptic neuron, from which it can be interacting with phosphate-activated glutaminase, which outputs glutamate and ammonia. Thus, the cycle will begin again. In the case of GABA, it's a little bit more complicated. When GABA is released to the synaptic cleft from the presynaptic terminals, one of two things can happen. Of course, it can be reuptaken from the synaptic cleft into the presynaptic neuron, or in the GABAergic synapse, GABA can be taken up by astrocytes and catabolized into the Krebs cycle intermediate succinate via GABA transaminase, an enzyme that is present in astroglial mitochondria. This GABA transaminase turns GABA into succinate semiiodide, and succinate semiiodide dehydrogenase turns succinate semiiodide into succinate. This succinate is converted to fumarate as part of the Krebs cycle due to succinate dehydrogenase. Then this fumarate is converted to malate via the enzyme fumarase. Malate is then converted to pyruvate by either mitochondrial malic enzyme or the cytosolic malic enzyme, and then the resulting pyruvate can be converted by pyruvate carboxylase, which is actually present in astroglial mitochondria as opposed in neuron mitochondria. The carboxylase turns pyruvate into oxaloacetate, and then other pyruvate that can be present in an astroglial cell can be synthesized into acetyl-CoA by using the three enzymes that make up the pyruvate dehydrogenase complex: pyruvate dehydrogenase, dihydrolipotransactase, and dihydrolipodehydrogenase. The products of acetyl-CoA and oxaloacetate is converted via an aldyl condensation and hydrolysis reaction that is catalyzed by citrate synthase. The aldyl concentration forms citral-CoA, which is then hydrolyzed to form citrate and coenzyme A. Citrate is then converted to isocitrate by aconitase using isocitrate dehydrogenase. This isocitrate can also be oxidized to oxalosuccinate, from which oxalosuccinate has its carboxyl group beta removed, after which alpha-ketoglutarate is formed. Aminotransferase transfers the alpha amino group to alpha-ketoglutarate to form glutamate, and from here the same process from the glutamate cycle occurs, from which glutamate is converted to glutamine via glutamine synthetase and can be transferred back to the presynaptic neuron. That was all for today's video. Thank you again for watching Neuron Idiot, and have a nice day.